Good evening, one and all. On behalf of the Department of Chemistry, St. Xavier's College, Autonomous, Kolkata, I hope that yesterday's session by Dr. Saurabh Chakraborty had enriched our audience to a great extent and hoping to have an equally amazing session on DNA, a Material World by Dr. Devleena Samanta this evening. Your enthusiasm and curiosity towards science had made us organize RACB'22 International Chapter 1, and today we have reached our second and final day of Chapter 1. So, without any further delay, I would like to commence today's webinar by calling upon our esteemed professor, Dr. Indranil Chakraborty, to introduce our speaker of the day.
Welcome, Devleena. Obviously, generally, I want to avoid this type of job, but this time I'm actually eager to introduce one of our student who is very close to my heart, and obviously one of your senior actually, none other than Dr. Devleena Samanta, currently an associate professor at University of Texas at Austin. Devleena did her schooling from Birla Girls High School, as far as I can remember. Then she joined our college, and in the same year, I joined the college also, 2007. And obviously, I can remember still that particular lecture in semester 1 on mechanism of vision by this particular group. Then she completed, obviously, the UG course in our college and joined Virginia Commonwealth University for PG course, where she did extensive research as a PG student, obviously, with Professor Jina, using extensive DFT calculation, and obviously there was several publications. And to mention, she had a JPCL on her own in 2014. Then she joined Stanford University with Professor Richard Neil Jade. Obviously, many of you know, probably many of you have a Facebook friend with this particular name. She had several publications in that particular group. Obviously, JAX, Nature Communication, Nanoscale, to name some of them. Then she moved on to Northwestern University and joined Professor Chad Merkin Group. Once again, pioneered some work on nanomaterials and biomaterials, that is, combining nanomaterials and biomaterials. And once again, got a wonderful publication there, AngioChem, Science Advances, PNAs, JAX, Advanced Material, to name some of them. And lastly, a Nature Protocol also. Currently, she is an assistant professor in the Department of Chemistry, College of Natural Science, University of Texas at Austin, working in chemical biology and material science. I hope this session will be a very much interactive session, because this is nothing but your senior with you. So with this introduction, I invite Devleena to RACB'22, Chapter 1, Day 2. Devleena, the audience is yours.
Thank you so much for that wonderful introduction. It is so nice to see all of you again. And let me share my screen. I hope everybody can see my screen right now. Thumbs up. Yeah.
Yes, ma'am.
Okay, awesome. All right, so let me start by saying, do you see what I see? I was hoping somebody would say I see SXC. I guess over Zoom, people don't really do that anymore. Anywho, so I would like to, get started by showing you some pictures. So I wanted to start with this really, this gem of a picture, I would say. And the reason why this picture is a real gem is because you will see that this is one of those rare pictures where you can see Icy smiling, but you can't really prove that Icy is smiling because, his son is blocking the smile. And then this is a picture of the chemistry lab as I remember it. And then in this lab, we had this, small room where you can see, this young and vibrant cohort of professors who we used to bother all the time. This is just a candid picture kind of capturing a moment like that. And then finally, I would like to end with a slightly more formal picture. And what you can see in this picture is our graduating class. So this was taken, I believe, the end of our 3rd year. And you can see some of the professors, not all the professors are pictured here, but some of the professors who really played an important role in shaping our future. So going to Xavier's really helped to shape my career. And I feel really privileged that I was able to go to Xavier's. So I just wanted to tell you that, you should all realize the privilege that you have, and you should take full advantage of the opportunities that are available to you. And so in terms of my research interests, in colloidal chemistry and nanomaterials was actually heavily inspired by Professor Chakraborty. I remember I used to teach us colligative properties at some point, and then at some point he gave me some papers by Munji Bawandi. And I read those papers. I didn't fully understand them at the time. I just remember thinking, oh, wow, that's super cool. And so that's kind of what I wanted to do. And then in my master's and PhD and postdoc, and now in my independent lab, that is sort of the flavor of research that I've carried on. And all of this is inspired by IC. All right, but you didn't come here to listen to my life story. You came here, to listen to the science. So I will move on to the science. And what I will do today is, most of my talk today is going to talk about DNA. Now, you probably know of DNA as being the building block of biology. But for the next 40 minutes or so, I want you to forget about that. Instead, I want to introduce to you a new concept where you can use DNA as the building block of materials. Okay, so what does that mean? So there's really 2 main areas that started developing sometime in the 1990s. One of them is called DNA origami. And the idea of DNA origami is just like you can, make different structures and fold paper into origami and make different shapes out of it. In the same way, you can fold DNA to make different shapes. That's the idea. So the way that you would go about doing this would be, let's say, you take a long piece of DNA, and what you do is you then fold it into the kind of shape that you want. And then you kind of hold these folds using short sequences that are called staple strands. So you kind of staple the folds together, and then you keep on going this way until you obtain the shape that you desire, which can be a square or a circle. And by simply following this principle, you can make shapes of arbitrary complexity. For example, you can make a smiley face that is made entirely out of DNA. So this is actually a microscopic image, an actual microscopic image of a smiley face that is made entirely out of DNA. So you can make something like this. You can also make something, a little bit more complex, like a map of the Americas. And all of this is made entirely out of DNA by folding a long piece of DNA, many, many times and holding it together using shorter staple sequences. So this is one way you can make materials out of DNA. Another way that you can make materials out of DNA was this idea was started by the Merkin group at Northwestern. And here the idea is as follows. You can take a nanoparticle. So a nanoparticle, is a particle that has at least one dimension that is less than 100 nanometers in length. So here in the example, what you can see, this gold ball is representing a gold nanoparticle. And what you can do is you can take short strands of DNA and densely functionalize the surface of the nanoparticle. So you're decorating the surface of the nanoparticle with short DNA sequences. So now you can use this DNA decorated nanoparticle or DNA functionalized nanoparticle sort of as a Lego. And the same way you can build different structures out of Legos, you can build different structures out of these nanoscale Legos. So you can organize these nanoparticles in three-dimensional space where they're kind of held together by DNA that acts kind of as a molecular glue. So here is an image of something like this, where you have these gold nanoparticles that are held together by DNA glue, and you can see that they form these crystalline shapes. So this is really the type of materials that is going to be the focus of my talk today. So this same structure, the DNA functionalized nanoparticle, this has two different types of applications. And depending on the applications, what we call these DNA functionalized nanoparticles changes. So in one application, you can use these as nanoscale Legos to build higher order structures. So in that case, we call these a programmable atom equivalent. You can also use this for biological applications, in which case we refer to the same structure as a spherical nucleic acid. All right. So in this case, you can use these nanoparticles, as chemical and biological probes, for example, or for therapeutic purposes. So same particle, two different names, depending on what our application of interest is. So let me start by telling you how, these DNA functionalized nanoparticles can act as programmable atom equivalent. So let's kind of focus on the name for a second. It's an atom equivalent. So it's not an atom, it's the equivalent of an atom. But unlike an atom, it is programmable. So I'm going to explain to you what this idea of programmable means. But I think central to this entire idea is we first need to really understand what about DNA is so special that it has really started this entirely new area or entirely new field. And to truly understand that, we first need to look at the structure of DNA. So here I've shown you the quintessential cartoon structure of the double helix of DNA. And if we were to focus in on a single strand, what we see is that your DNA has some sugar, some bases, and some phosphate groups. And we know that these bases have very specific interactions. So we know that, T binds with A and then C binds with G. And this is what gives DNA what is called its programmability. The meaning of the word programmable here means that if I were to design a sequence of DNA, it will only bind to its complementary sequence. So I can kind of program what sequence the DNA binds to by generating a sequence that's complementary to it. When we think about building materials, I'm going to talk to you about how we can make materials out of DNA. But really the first thing that comes to your mind when we think about materials is the periodic table, right? And that is because the periodic table is basically a menu of every possible building block that there exists. And all materials are nothing but combinations of, one or more of these elements. Now, something that you have learned in your high school chemistry and also in your undergrad chemistry is that the properties of materials depend on their composition and structure. All right. So composition would mean what the chemical composition is. If you have table salt, for example, that is salty versus sugar is sweet. And that is because the chemical structure is different, right? So your composition, and structure are different. So composition really refers to what atoms are connected together, whereas structure actually refers to how these atoms are connected together. For example, you can have the same composition. So you can have, let's say, different allotropes of carbon, right? So you can have graphite, where we know it has a sheet-like structure. Or you can have carbon in the form of diamond, where it has this tetrahedral geometry. And what you will find is, even though the composition is the same, just because the structure is different, you see completely different properties. Whereas graphite is soft and slippery, you will see that diamond is actually the hardest substance that's known to man. So the structure tells you how these atoms are connected together. So once again, from our high school chemistry knowledge and also from your undergrad chemistry knowledge, what you know is that the properties of materials depend on the composition and structure. But turns out that when we go to the nanoscale And once again, just to remind you, the nanoscale is when one dimension of a material is less than 100 nanometers. So here in this image, I've kind of pictorially shown you different length scales, right? So if you have like a gems, that is going to be about 1 centimeter, human hair is about 100 micrometer. And then you have bacteria somewhere at 1 micrometer. And then, viruses and proteins start to get into the nanometer length scale, and where 1 angstrom is really the diameter of a hydrogen atom, right? So somewhere between a hydrogen atom and a virus is where your nanoparticles lie. So when you come to this nanoscale, this new size regime, what happens is the properties of materials are not only depend on composition and structure, they also depend on size and shape. All right, so what does that mean? So let's think about this for a second. So if I were to, tell you that I'm going to make materials out of silver, so I can make silver jewelry, I can make a silver bar, or I can have a silver coin, and they all look silver, right? They all pretty much have the same properties. They look the same, for example, color is same. But if I wanted to make different shapes of silver at the nanoscale, so if I were to make silver spheres or cubes or plates, or as you can see on the right, silver nanowires, then what you will see is that the properties, for example, in this case, the color, will change dramatically as you change the size of your nanoparticles and the shape of your nanoparticles. So you can see like pretty much you can get like all colors of the rainbow simply by changing the size and shape, right? So this is not something that we see in bulk matter, but this is something we see in on the nanoscale, right? So this is one example of things that change at the nanoscale. All right, so now that we know that properties can be different at the nanoscale, now this brings us to this concept of then what is then this programmable atom equivalent. So a programmable atom equivalent, as we talked about previously, you have a nanoparticle core, and on that nanoparticle, you decorate the surface with a dense shell of short DNA sequences. And these DNA sequences end in a single-stranded region that is called the sticky end, all right? Very simple and intuitive nomenclature, sticky end because it sticks to things, right? Okay, so now imagine I have 2 different sets of nanoparticles. Let's say the nanoparticles are identical. The only difference is that they have complementary sticky ends. What's going to happen is if I were to put these nanoparticles together, they're going to come together and form a DNA bond between them. So the same way atoms come together and form bonds, I can make nanoparticles come together and form and be stuck together through DNA-mediated bonds. All right, so your nanoparticle is now acting like an atom, so it's an atom equivalent, except as it is programmable, because by changing the length and the design of your DNA sequence, you can actually program what other type of nanoparticle it binds to, and consequently, what the overall structure is. This is something that we cannot do in atomic systems. Carbon will have a valency of 4, and it can only form certain types of structures. Whereas for these DNA functionalized nanoparticles, by changing the shape and size of the nanoparticle, as well as the DNA design, we can make this form a wide variety of structures. Okay, so the same way atoms come together and form like higher order materials like crystals, your nanoparticles can come together and form colloidal crystals, where your nanoparticles are colloids. And here is an image of a crystal like this. So this is a crystal that is made entirely out of gold nanoparticles. And this is a scanning electron micrograph of a crystal like this. And you can actually see the periodic assembly of your nanoparticles. You can see where the nanoparticles lie, and the gap that you see in between them, that's basically occupied by DNA. How cool is that? Okay, so as I said, unlike atomic systems, you can actually change the shape of the nanoparticle. So you can change the shape of your atom, basically, right? You can change the composition of your nanoparticle, and then you can change your DNA design. And by doing so, you can access a wide range of structures. So over 500 different structures have been made using these nanoparticle DNA constructs. So then, when I started working in this area, one question that I wanted to ask is, all right, so we can make these structures. Can we make flexible structures? So when you think of a crystal, like a salt crystal or a sugar crystal, you really think of it as something stiff, right? So the idea is that we have DNA bonds between these nanoparticles. Can we make our crystals squishy? So you apply some stimulus and your crystal shrinks, and then you apply some stimulus and your crystal grows, something like that. Can you do that? And because, DNA is a soft material, I figured that maybe there could be some way we could do that. And so this is, on the left, what you will see are a real-time microscopic video of what is happening to your crystals as you add, for example, a high concentration of multivalent cations. You're adding a high concentration of salt, and you see these crystals are shrinking. And because we can't really look at the structure of the DNA by experimental methods, on the right, what you can see are molecular dynamic simulations that show you what is happening to the DNA structure as these crystals are shrinking. So this is another thing that you can do with DNA-mediated colloidal crystals. You can also make them flexible, where you have control over a fourth dimension, not just 3-dimension, how you arrange your nanoparticles in space, but also over time, how they change. So you can make flexible and dynamic materials with DNA. Okay, so why would I care about this? Because this entire idea really allows you to make nanoparticles from make materials from the bottom up by organizing nanoparticles in 3-dimensional space with complete control over the interparticle distances. So now you can make materials and then study distance-dependent properties of these materials. So, you can imagine having like a smart window where, let's say, you have this exact same material, but when you change the distance between the nanoparticles, maybe it is transparent. And then when you change the distance between the nanoparticles, the same material becomes non-transparent. So you have like a smart window that is or isn't transparent depending on the interparticle distances that you have. You can study substrate channeling in catalytic systems. You can have, study magnetic properties by changing interparticle distances, or you can imagine making mechanical metamaterials. So a metamaterial is a material that doesn't exist in nature, and it has some property that is not found in nature. So it's an artificial material with some desirable property that you won't really find in nature. So we can start to make these kind of cool things all the way from the bottom up by design. So here is one example of something like that, of what you can do with this flexible DNA bonds, right? So here is a structure. So what you see here is you have a surface, and on this surface, you have the blue cubes are representing gold nanocubes. So on the surface, you have gold nanocubes that are stuck to your gold surface with DNA. Now, you know what reflection is, right? Reflection tells us that angle of incidence is going to be the same as the angle of reflection, right? Okay, so that is what you see here. If you can see my cursor, what you see is, if you have the surface with a particular DNA length, when light hits, you see normal reflection. But now, because these DNA bonds are flexible, once again, I can apply some stimulus that will shrink these DNA bonds. And when these DNA bonds shrink, even though you have the exact same structure, the only difference is that the distance between your gold nanocubes and your surface is changing. It's becoming smaller. Now you see something called anomalous reflection, meaning when you hit it with a beam of light, you no longer see reflection at the angle of incidence. You actually see the light getting reflected at a different angle, right? So you see this kind of like weird behavior that you would not really find in natural systems. So this is the first example of a two-state optical metasurface. So you can have the same material that you can toggle back and forth between two different functions simply by changing the interparticle, simply by changing the distance between the surface and your nanocubes. Okay, so what are the take-home messages that we learned so far? So I hope I've convinced you that DNA is really a powerful material building block, and you can use it sort of like a ligand as you use in inorganic chemistry, and you can direct the organization of functional materials into 3D macroscopic structures. These DNA bonds are tunable, so you can change your DNA length. They're flexible, and then you can make materials with properties that you do not see in nature by using DNA-mediated assembly, right? So this is why this area is so important and has really sort of transformed the area of material science. All right, so these are all examples of how you're using this DNA functionalized nanoparticle to build materials. Now, you can take the same material and now think of this material from a biological standpoint. So in a biological context, you know that there is linear DNA, so that's just a DNA sequence. There's circular DNA, so that's something like a plasmid, so the DNA that you would find in bacteria, for example, that would be circular DNA. So in the context of biology, this structure obviously looks very different from the natural forms of DNA that you might imagine. And to distinguish it, it's called a spherical nucleic acid. So it's the exact same structure that we were calling a programmable atom equivalent. But just because our application now becomes biology, we call it a spherical nucleic acid so that we can distinguish it from naturally occurring forms of DNA. And in the context of biology, you can think of this particle sort of as a multifunctional tool, right? So like a Swiss knife. So by changing the DNA sequence, you can change the application. So you can detect different molecules in cells, or you can treat different diseases just by changing the DNA sequences. So I'm really going to focus on the detection part because that's what my research is focused on. So I think the first question that comes to mind is that, all right, so I said that you can use this, as chemical and biological probes to detect different molecules. Why is it so important to detect different molecules? So I'm going to say that not only do we want to detect things, we want to detect things in living cells. And looking at things in living cells is a grand challenge. So, as a chemist, if you wanted to look at the chemical composition of a cell, of a dry cell, so minus the water, what you see is that, your cell is made out of proteins, some nucleic acids, some ions, some lipids, sugars, so on and so forth. Now, there are very good ways of looking at proteins in living cells. For example, you can express a protein with a fluorescent protein. So you can tag it with a fluorescent protein, and then wherever this protein goes, your fluorescent tag moves along with it, so you can really learn a lot about what this protein does, right? Obviously, this was a very, very important development, and it was awarded a Nobel Prize in 2008. There are also very good ways of looking at nucleic acid in cells. So nucleic acids are DNA and RNA, not necessarily of looking at nucleic acids in live cells, but there are ways of looking at nucleic acids in cells. Examples are polymerase chain reaction, so PCR. You probably have heard of PCR in the context of COVID detection, where you can amplify your DNA and RNA targets. And obviously, this is also a very, very important development, and it was awarded another Nobel Prize. But if you were to look at this right side of the circle, these ions, lipids, sugars, so on and so forth, these are called the dark matter of cells. And that is because it's really, really hard to detect them or see them in living cells, all right? And if we can look at things in living cells, we can really start to understand how these molecules play a role in cellular development and how the dysregulation of these molecules leads to diseases. Okay, so then the question that we wanted to ask is, can we develop a general class of probes for live cell analysis? So something that can not only detect proteins and nucleic acids, but can almost detect anything, in the cell. That was the question that we wanted to ask. So I want to create a probe, and we don't really know what the structure of the probe is, right? So my probe structure is a black box, right? But the idea is that this probe needs to be able to enter my cell, my living cell. And then if my molecule of interest is present in the cell, so this molecule of interest is called a target. So if my target is present in the cell, this probe will give me some sort of signal. And if it is a fluorescent signal, then it's a really, really easy way of knowing that my molecule is present, my target is present, all right? So that's what we really want to do. So what are the criteria of making a probe like that, right? So how do we kind of figure out what this black box looks like? Well, first of all, if you want to look at things inside cells, your probe needs to be able to enter your cells. If it's entering the cell, it should also be stable inside the cell. It should result in a signal when it interacts with your target. It should be specific. So if you're looking for glucose in the cell, it shouldn't light up in the presence of other sugars. It should be sensitive, meaning even if we have small quantities of the target molecule that we want to look at, we should be able to detect it. And then it should be biocompatible. So it should be something that doesn't kill your cell, right? And finally, this would be a really added bonus if this probe were also modular and generalizable. So what does modular mean? So imagine, you make a car, right? And you want to make a car that goes on flat road, regular road, a car that goes on sand, a car that goes on snow. Every time, you don't really want to make a car from scratch. Ideally, what you would like to do is you would like to make the framework of a car, and then you would switch out different parts. Like you would switch out the tires so that the same car can go on flat road, the same car can go on sand, the same car can go on snow, right? So this means modular. You can just switch out parts and change the function, right? So if we can do this, we don't really have to make a new molecule from scratch every time. And then our probe would really become generalizable. So these are some of the things that we would like to have in our probe. Okay. So how can we do that? Well, the first thing we need is something that can interact with a wide range of molecules so that we can, detect them. And turns out that nucleic acid-based structures can bind to various molecules. For example, if you have one strand of DNA, as I mentioned this before, if you have a single strand of DNA, that can bind to complementary nucleic acids. So if you have a single strand of DNA, you can design it so that it binds to, let's say, an RNA present inside a cell. Moreover, you can evolve DNA sequences, and these DNA sequences are called aptomers that bind specifically to ion small molecules or even proteins. So here I've shown you the structure of one such DNA sequence that binds to 2 molecules of ATP. And then that allows you to look at non-nucleic acid targets. So things like ion small molecules and proteins. So you can really think of an aptomer as an artificial antibody. And instead of being made out of amino acids, so instead of being made out of a protein, this artificial antibody is made out of DNA, right? So bottom line is you can design DNA sequences that binds to both nucleic acid and non-nucleic acid target. So if my aptomer, let's say I have an aptomer that binds to my target molecule, if this binding somehow leads to the generation of a signal, and once again, preferably a fluorescent signal, I will know that my binding has happened, and that would tell me that my target is present. So what I need to do is I need to figure out a way that generates a signal when my target molecule interacts with the aptomer or the artificial antibody that I've made for it. Okay, that's what I need to do. So that's what we did. So in my postdoc, what I did was I developed a new class of signaling aptomers called forced intercalation aptomers. So the idea here is that you have this piece of DNA, and by itself, this piece of DNA is kind of like a floppy little molecule. And this aptomer has a dye attached to it. Okay, this is called a forced intercalation dye or a FIT dye. Okay, now when this aptomer binds to the target molecule that it's supposed to bind to, this floppy DNA strand adopts this complex secondary structure. And as a result, this dye is forced to intercalate or fit between DNA base pairs. And when this dye fits between DNA base pairs, its fluorescent signal turns on. So that's how this works, right? So that means that if I can design these antibodies or artificial antibodies that basically just light up in the presence of the target molecule. So that's what we did. And we showed that we can, this is a very, very simple design. And with this, we were able to detect a wide range of different targets. And I would not really go into the details of this right now, but we also showed that this has several advantages over existing techniques of how you can detect molecules using aptomers. But one cool thing that we were able to do is we showed that you can detect a steroid hormone in clinical samples like human serum, right? So detecting things in buffer is one thing. Detecting things in complex biological media is entirely different because you have, thousands of other molecules that can interfere with your measurements. And what we found is that using this new fit aptamer that we had designed, we could look at these steroid levels. And this steroid particularly, it's called DHEAS. This steroid is associated with stress levels, and it is also associated with adrenal tumors. So here, if you focus on this middle chart over here, what you see is in black, you see what is called a control, meaning you add an aptamer that doesn't really result in a turn-on signal upon binding to DHEAS. And what you see is that the signal doesn't change even if the concentration of DHEAS is changing in your sample. But then when you add an aptamer that lights up in the presence of DHEAS, as you increase the concentration of your DHEAS, you get more and more signal. All right. Okay. So this then tells us that we have a quantitative way of looking at DHEAS in clinical samples. And we went on to show that we can detect a wide variety of analytes. We can detect ions, we can detect proteins, and we could design fit aptamers or forced intercalation aptamers for every aptamer target binding mode that's been reported in the literature. Okay, so this works really well in buffer. I have talked to you about things working in buffer. I've talked to you about things working in complex biological media like serum, but I've still not talked to you about looking at things inside living cells, right? So what is the problem here? The problem is DNA sequences on their own do not enter cells because your cell has a cell membrane that is negatively charged and it prevents the entry of nucleic acids into cells. Secondly, you really should not have DNA floating around in your bloodstream or RNA floating around in your bloodstream. That happens when you have an infection, like a viral infection or a bacterial infection, which means that your body is programmed to destroy any DNA that comes into your body. So it has these enzymes called nucleases that will rapidly chop up DNA. So if I wanted to detect things inside living cells with linear DNA sequences, they're going to be rapidly chewed up, right? So I have a stability problem and I can't even get inside the cell very efficiently, right? So I can't use these fit aptamers on their own to detect things inside cells. This is where, once again, the DNA functionalized nanoparticles come into play. So these spherical nucleic acids, I'm calling them spherical nucleic acids because now we're talking about biology. These structures have emergent properties. What is an emergent property? An emergent property is a property that you don't see in constituents, but you see in the overall structure. For example, so the way you make the spherical nucleic acid is you have linear nucleic acids, you have a spherical nanoparticle core, and you put them together to make your spherical nucleic acid. So the linear nucleic acid or the gold nanoparticle doesn't really enter cells. You make this structure, and this huge structure turns out enters cells in really high quantities. So you have a property simply by virtue of structure that you do not see in any of the individual components of the structure. These structures also bind to complementary nucleic acid sequences more strongly, meaning we can do more sensitive detection. Also, this arrangement makes your structure much more stable with respect to enzymatic degradation. So, you imagine you have like this forest of trees. So if your, enzyme comes in, it's going to be harder to access an individual tree and cut the tree down. Whereas if you have the linear nucleic acid, it gets rapidly chopped up. So you have increased stability, you can enter cells, you don't really see a high immune response, and it can bind to complementary nucleic acid sequences more strongly, meaning you can detect things more sensitively. So you really have now the black box that I showed you before, the structure of the probe, you really have figured out what you can use to make probes. So based on this was the first platform that allowed one to look at the genetic content and metabolic content of living cells. So based on a spherical nucleic acid, so which is a gold nanoparticle functionalized with DNA sequence, the first platform that was built was called a nanoflare. All right. So this is a probe that is based on the spherical nucleic acid architecture. So what you have is, you have a gold nanoparticle, and on this gold nanoparticle, you have DNA sequences. We call these DNA sequences the recognition strand. So these are the strands that are designed to bind to whatever target molecule that you're interested in looking at. To these recognition strands, we have short fluorophore labeled sequences that are hybridized. These are called flare strands. So your gold nanoparticle acts as a quencher, meaning when your fluorophore is hybridized here, you don't see any fluorescence. The fluorescence is off. Now, when your target molecule comes in, let's say it is an mRNA that's present in a cell or messenger RNA, it can bind to your recognition strand and in the process displace your fluorophore labeled strand from the surface of your nanoparticle. And because your nanoparticle and your fluorophore are no longer in close proximity, you get a fluorescence turn on. So now you get signal because of this recognition event. So what you can see here is that now I can treat cells with nanoflares and I can look at their genetic content. So here on the left, what you can see are cells that express survivin. So this is a gene. It has been implicated in many forms of cancer. So when you have cells expressing survivin, you see that they light up, right? You see a lot of red fluorescence. But when you treat cells with a nanoflare that doesn't bind to survivin, you don't really see as much signal, right? So these cells are expressing survivin, but you treat it with a nanoflare that doesn't bind survivin, you don't really see a lot of signal. So this tells you that you can make probes that are relatively specific. Okay, so that's all good. But the problem is your signal is dependent on the distance between your fluorophore and your quencher, which is your nanoparticle, right? So that means that I can get a false positive signal in many ways that has nothing to do with my target finding my nanoflare, right? So if I go back here, what you see here is, you don't see as much red in this right picture as you see on the left, but you still see some red. So you don't really know if you have lesser signal or if this is a background signal. So the reason that you see this is because, like I said, you have these enzymes that can still degrade these structures. And as a result, you can change the distance between the fluorophore and the quencher. Your flare sequences might bind to proteins inside the cell and just fall off your nanoparticle. Once again, you'll see false positive signal. And then finally, your cell has a very reducing environment. So the way in which you attach your DNA sequence to the gold surface, that bond might get broken inside the cell. And once again, you'll see a false positive signal. So I think the question then becomes obviously the limiting problem here is that our fluorescence is dependent on distance. So can we develop a strategy that is not dependent on distance? So can we develop a strategy that doesn't really need a quencher? And that is where this fit strategy can come into play. In the fit strategy, you don't have a quencher. Your dye is non-fluorescent when your sequence is not bound to the target, and it only fluoresces when your sequence binds to the target, right? So based on this fit aptomers, we then build the next generation of nanoflares, which are called protein spherical nucleic acids, all right? So in this case, let's look at what the structure looks like. So instead of an inorganic nanoparticle core, we have a protein as the nanoparticle core, right? So you can think of a protein as an atomically precise nanoparticle, right? So here, your nucleic acid shell allows this whole thing to go inside the cell. And you can use fit aptomers to detect different molecules of interest. And then using the protein core, because you don't need a quencher, you don't need to use gold anymore, you can use a protein. That really allows you to site-specifically modify the protein. So you can attach multiple things at different sites on the surface of the protein. And moreover, there are many different targets for which DNA sequences or DNA recognition sequences are not known. For these types of targets, you can use the protein's recognition capabilities to detect these molecules. So things that you can detect with DNA, you can detect with protein. So this really expands the scope of recognition of targets. Okay, so we wanted to show proof of concept, right? We wanted to show that we are making this structure and that it functions the way that it's intended to. How did we do that? So we made a fed aptamer that would change its structure in response to changing pH. So let's say you have a fed aptamer. So this is made with a DNA sequence called the I motif. So this DNA sequence is basically a bunch of cytosins. When you reduce the pH, this type of DNA sequence can form a quadruplex structure. And so if you replace one of the bases with your dye, then the dye will be forced to intercalate between your cytosine bases and it'll turn on. So what you would see is if you have the structure at a higher pH, it would be non-fluorescent. And then at a low pH, when the I motif structures are formed, you would see high fluorescence. So that is sort of the idea behind this. So then what we did was we treated some cells with our structures, and then we clamped the pH, meaning we fixed the pH of the cell at different values. And then we analyzed thousands of cells, one single cell at a time, okay? So this could be done by a technique called flow cytometry. So you can look at one single cell at a time and you can look at thousands of cells at once. And what we found is that the cells that are at a lower pH are twice as fluorescent compared to the cells that are at a higher pH, which agrees with what we were expecting to see. But the really cool thing is you can now also use the protein's functionality, the protein core's functionality, to detect molecules for which there are no known DNA sequences. And to show this, what we did was we developed a new assay for looking at glucose inside cells. And to do this, we chose glucose oxidase as the protein. So here what you can see is this is what glucose oxidase does. In the presence of glucose, glucose oxidase will convert glucose into gluconic acid and hydrogen peroxide. So if you have a dye that fluoresces in the presence of hydrogen peroxide, then you're actually, indirectly measuring glucose, all right? So that's what we did. So we treated our cells with the glucose oxidase spherical nucleic acids, and then we measured fluorescence. Okay, so the first thing we did was we just took these glucose oxidase spherical nucleic acids and just treated these with different concentrations of glucose in buffer. So no cells yet, all right? So just in buffer, we see that the fluorescence increases by up to 120-fold in the presence of glucose. And the signal is very, very specific for glucose over other sugars. Finally, when we treated our cells with glucose oxidase, this is what we see. So let me show you how to read this kind of a graph. So in this graph on the right, what you see is, this is showing you a histogram. So it shows you a fluorescence histogram of cells. So what you see here is on the x-axis, I'm plotting fluorescence. And then on the y-axis, I'm plotting how many cells have that particular fluorescence, right? So this is a histogram. So in black, what you see are cells that are untreated. So not very fluorescent, right? Because your cells have a very low fluorescence. When you treat your cells with this hydrogen peroxide sensitive dye, you see that the fluorescence increases. So the entire population shifts to the right, increased fluorescence. And that makes sense. Your cells do make some amount of hydrogen peroxide, and that is exactly what you're seeing here. But now when you treat with your cells with the glucose oxidase spherical nucleic acids, you see that your fluorescence shifts by up to 12-fold. And this extra fluorescence that you're seeing is because of the extra hydrogen peroxide that's being generated because of the glucose, right? So we are able to detect glucose intracellularly. So now you can do some cool things with this new probe. For example, if I were to, let's say, feed my cells different concentrations of glucose. So let's say I don't feed my cells glucose at all, and I feed my cells 25 millimolar glucose. I see that the cells that have been fed glucose are more fluorescent than the cells that were not fed glucose. I can also feed my cells the exact same amount of glucose, but I can block the transport of glucose into the cell. So I can block the entry of glucose. And then I see that the cells that have been treated with these blockers are not as fluorescent as the cells that can take up glucose. And finally, if I were to treat my cells with insulin. So insulin promotes the intracellular uptake of glucose. I see that my cells become more fluorescent. So bottom line is, I can use these structures for high throughput drug screening, right? So I can treat my cells with different molecules and see if the glucose levels inside the cells are changing or not. So I can use these to screen drugs in high throughput. So, at the beginning of this part of the presentation, I asked, can we create a general class of probes for live cell analysis? What we see is that these new protein spherical nucleic acids actually satisfy all of the requirements that we have, right? They enter cells, they're stable, they result in a signal, they're specific, they're sensitive, they're biocompatible because they're made out of proteins and DNA, biological materials. And finally, they're modular and generalizable. So you can just switch out the DNA sequence with another sequence that can detect a different molecule, and consequently, you can detect something else. You can switch out the protein to a different protein and detect a different molecule. So the take-home message is that protein spherical nucleic acids really represent a plug-and-play platform, right? So you switch out sequences, you detect different things. And you can detect things through binding-based sensing that is enabled through the DNA. You can also detect things through activity-based sensing. So when you're looking at glucose, your protein is not really binding the glucose. It's acting upon the glucose that's resulting in a molecule that we are looking at. So we are doing activity-based sensing here. And the sensitivity that we obtain in these measurements is at par with ELISA. Okay, so at the beginning, I mentioned PCR, which you have heard of in the context of COVID, and you've also probably heard of antigen testing, right? So most of these antigen testing rely on the principle of ELISA. So these are antibody-based signaling techniques that allow you to look at very, very small amounts of material, and our probes are able to give you the same kind of sensitivity, except we are doing things inside cells. You don't have to take a bunch of cells and grind them up. So really, these protein spherical nucleic acids have opened new opportunities in chemical and biological sensing. All right, so overall, I told you about these DNA functionalized nanoparticles and how you can use DNA to make different types of materials. In one area, in one application, you can use DNA as a ligand to organize materials into higher order structures. And in the other, you can use DNA to make spherical nucleic acids for biological purposes. So this work was done. All of the work that I presented today, for this, I would like to acknowledge my advisors with whom I worked, and all the wonderful scientists and colleagues and graduate students, postdocs, and undergraduate students, and even high school students who contributed to this research, as well as the funding that supported this work. Okay, so in the last few minutes, I would like to tell you a little bit about what my independent laboratory aims to do. So my lab is at the interface of analytical chemistry and synthetic chemistry, and we want to address challenges in biology and medicine. So what I really want to do is I want to use chemistry to impact human health. And as a chemist, when I look at the human body, what I see is that it is really, there's some 37.2 trillion cells in the human body. And there are millions of chemical reactions happening in every cell. So let's say you can write out each reaction, right? So you can write out all of the individual reactants and each of the individual products. Now imagine we can monitor or detect each of the individual reactants and each of the individual products that are associated with these chemical reactions. We can measure or quantify each of the individual reactants and each of the individual products associated with these chemical reactions. And if we can manipulate or control each of the individual reactants and each of the individual products associated with these chemical reactions, then in principle, we can detect and treat any disease. Now, of course, it's not as simple as that sounds, because for this, what we need to do is we need to make new materials and we need to make new methods. So that is what my lab aims to do. We are at the interface of analytical chemistry, chemical biology, and material science. And we develop new materials and methods to monitor, measure, and manipulate biochemical processes. And our ultimate goal is to develop improved diagnostic and therapeutic platforms. Toward this end, what we are working on right now, we are working on structures that get activated in specific cell types so that you can detect a molecule of interest, or you can treat a specific disease by degrading a specific molecule of interest. We're also trying to look at protease activity associated with cells. So proteases are basically proteins or enzymes that degrade other proteins, and they're heavily implicated in various diseases like infectious diseases, cancer, so on and so forth. And what we want to do is we want to build the first platform that allows us to look at many different proteases simultaneously with single cell resolution. And we believe that by doing this, we will be able to provide comprehensive fundamental insight into how protease activity is dysregulated in diseases. And we hope that this will allow us to detect diseases much earlier than what's possible now. And then finally, on a more materials chemistry side, we try to make structures where we can precisely control the position of enzymes in three-dimensional space. So, if you look at nature, you see that all the functions in nature that are mediated by proteins or enzymes are really comprised of multi-protein complexes, where the proteins are arranged in very specific ways. So if you think about photosynthesis, you have a bunch of different proteins. They're arranged in a very specific way that enables photosynthesis. We want to be able to do that synthetically, where we can organize enzymes in three-dimensional space. And this is my group at UT Austin, and my group members are really going to carry out my vision forward. And I'm very grateful that I have this vibrant group of students working with me. And then finally, I would like to end by saying that please feel welcome to apply to UT Austin. Applications to graduate school open in fall 1 year in advance. So if you were to apply for 2024, you would be applying, or 2023, you would be applying in 2022 fall. We don't have any GRE or TOEFL requirements. We are ranked 4th in analytical chemistry in the US, and we are ranked 22nd most productive university in the world. Right? With this, we come to the end of my presentation. Thank you so much for your attention, and I would be happy to answer any questions.
Thank you, Devleena, for this wonderful lecture full of new philosophy. And obviously, right now the session is open for question, and obviously she will be very much interested to answer your question, and obviously, if possible, to take some question to her lab also.
So I see a question in the chat. I think it says, what metals or organic compounds we use to synthesize spherical nanoparticle cores in SNAs? So you can actually use any, like in principle, you can use any material. So typically gold is used because it's very easy to attach thiolated DNA to that. But as I've shown you, you can use a protein core. People have used MOF cores. People have used polymer cores. People have used lipid nanoparticles. So you can use a wide variety of materials as the core, almost any core that you can think about.
Thank you, ma'am. There are a few questions from our audience. Is there any possibility that the DNA nanoparticles will reprogram neurons, making them undergo mitosis for regeneration?
Sure, yes. Yeah. So you can design DNA sequences, so that they perform a particular function. So one thing that you could do is like, it has been shown that you can use these DNA sequences to do gene knockdown, for example, right? So you can also imagine that you can do gene knock-ins and stuff like that. So you can promote or suppress, different molecules by just simply changing the DNA sequence that you're using.
Thank you, ma'am. There is another question from our audience. What exact changes occur in the SNAs that allow them to easily permeate cell membranes?
So that is a really good question. And, whenever somebody says that, that means that they don't know the answer. And the truth is that this is not very well understood. So what we do understand, or what we do know, is that these spherical nucleic acids enter cells through receptor-mediated endocytosis. So there are these scavenger receptors on the surface of cells that allow these highly negatively charged spherical nucleic acids to enter the cells. But if you think about, let's say, a plasmid DNA, right, a bacterial DNA, it has a lot of negative charge on it. That doesn't enter your cells very well, but these spherical nucleic acids do. It is not at all well understood why SNAs enter cells over these other very highly charged species. It's not understood.
Thank you, ma'am. There is another question. How are the DNA bonded to the nanoparticles, as in what forces are acting between them?
So the DNA is chemically conjugated. So if you have a gold nanoparticle, you can use thiolated DNA, so you'll have a gold thiol bond between them. If you have, let's say, a polymeric nanoparticle, you could put, let's say, azides on the surface of your polymer, and then you can add alkyne tagged DNA, that you can attach through click chemistry. So it is all covalent bonding that's happening between the DNA and the nanoparticle. And what chemistry you would use will be dictated by the surface chemistry of the nanoparticle that you have.
Thank you, ma'am. The last question we have is, can we produce new eco-friendly materials using this technique?
What do you mean by eco-friendly materials? Like how? How so? Like what? I'm not sure I understand the question very well.
Okay, ma'am. Thank you so much for answering all our questions. Now, I would like to call upon our professor, Dr. Indraneel Chakraborty, to deliver the official vote of thanks.
Thank you once again. And obviously, this is official, so I need to thank our principal, Father Dominic Savio, Vice Principal, once again, Bertram De Silva, for their help, the entire department, the students, and obviously Devleena Yo. And for me, it is more than chemical biology and material science. It is actually something engineering using chemical and biological molecules, what you are doing right now. So wonderful work, wonderful view. Wish you very many wonderful work in your near future. And obviously, if whenever you come to Kolkata, obviously, please visit our department, your own department.
Will do.
Thank
will
you.
do. Probably next month.
Thank you. Thank you.
Thank you so much. It was wonderful to see you again.
Thank you. So with this, we will end today's program. Thank you, Devleena, once again.
Thank you so much.
Thank you, sir. With these words, we come to the end of RACB'22 International Chapter 1. Hope all of you have enjoyed and learned a lot from these webinars. I would request all the participants to fill up the feedback form rolled out in the YouTube live chat within stipulated time. We will be back again with RACB'22 International Chapter 2 on 24th and 25th of February 2022. Till then, subscribe to our YouTube channel for getting further updates. Have a nice weekend ahead. Thank you.